So here we are with Rebecca Shapiro from University of Guelph. Thank you very much for accepting the invitation for this interview. And I'm going to start with the questions.
Great.
So first, what is your area of research?
So my lab at the University of Guelph, we work on microbial fungal pathogens. So we study fungal pathogens that cause infection mostly in humans. One of our big areas of research is on Candida, which is a yeast pathogen and causes sort of a range of disease from more kind of benign superficial mucosal infections to more severe, invasive, life-threatening infections in immunocompromised patients. So we study Candida, and we use functional genomic techniques to sort of understand some of the basic biology and pathogenesis of Candida. And we develop CRISPR tools is sort of a big part of our research program. So we sort of develop and optimize new types of CRISPR technologies that we can apply in Candida and other fungal pathogens, and then use these to, sort of alter and manipulate the genetics of these organisms and study their biology in different ways.
Perfect. Thank you very much. Very interesting. I've seen in Twitter some of your publication and a deposit. I think your lab is sharing a lot of these tools with Adgene. I've seen.
Yep.
So that's
Yeah, definitely a great part of sort of science right now, I think, is the I don't know, the collaboratives, open science sort of part of it. And boy, yeah, Adgene as a way to sort of deposit all the new tools you make. We just love that. And we've had a lot of people, get our plasmids, our new CRISPR tools through that. So that's very rewarding, actually.
Perfect. So, second question, how do you describe your research to the members of the public?
So yeah, probably kind of similarly, but yeah, we study fungal pathogens. These tend to be sort of lesser studied than some other human-associated pathogens. Obviously right now, I think the only thing on anyone's mind is viruses. Otherwise, we often think about bacteria, but we don't think about fungi as much. So those are the pathogens that we're interested in. And yeah, we're interested in studying their genetics. So we're designing tools that we can use to knock out different genes in these pathogens one by one, and then study them to see if knocking out those genes affects their ability to cause disease. And if so, then we've just found something interesting and we can start to kind of dig in and figure out how that gene is responsible for, for causing infection or maybe for allowing pathogens to survive in the presence of antifungal drugs. It's another one.
Yeah, very interesting because I remember in the last CSM meeting, I was talking to another scientist that also worked with yeast, and he told me that nobody cares about microbiome studies with fungi, and he was trying to do that, and now that you mentioned that not many people work with.
Yeah, it's true. Fungi are sort of left out of a lot of microbiology initiatives, and the microbiome is a big one. And we bias it very heavily because when we do microbiome studies, we only look often, I should say, only look for bacteria and just, you only find what you look for. So,
yeah.
Yeah.
So the third question, do you remember that moment that make you want to study microbiology?
No, because I don't think there was a moment. I think I think I fell into microbiology by accident. And I'm right now, as just earlier today, I'm teaching Introduction to Microbiology second year microbiology class at the University of Guelph. And as I told my students, I've never taken a microbiology class in my life. So I did an undergraduate degree in biology, which was really general. It was like developmental biology, genetics, molecular biology, did not take a single microbiology class. And towards the end of my undergrad, I took one of these upper level seminar classes for fourth year students where there's only a couple students and you read research papers, and that was so exciting. And one of the research papers I read was by someone who had just started a faculty position at the University of Toronto, Leah Cowan. And I was so excited and I wanted to go work with her to do my PhD. And that's what I did. And then I sort of fell into becoming a microbiologist through that, essentially.
Interesting. It's similar with me. I don't think I never wanted to be microbiologist.
Yeah.
Because I studied biochemistry and then I discovered that was very thin the difference between biochemistry and microbiology because you need a model,
Exactly.
no?
Exactly. I think for me, it was more genetics. Like I love genetics and evolution and these sort of things. And yeah, I think I fell into that. And this was the model. We were using that in fungi. So that's.
And it's interesting that you mentioned that you decided to go to a new faculty professor, and that was really good for you.
Yeah, yeah, I think I people wonder about that often, I think, right? It's sort of a risk benefit thing to starting off with someone who's just starting their own lab. I guess now I am biased because now I run a pretty junior lab and I think it's a great environment to work in. I don't even think it occurred to me as a new graduate student that that was maybe a risky thing to do. I think I thought the research was amazing and the lab, was amazing. And I think I just dove in.
Interesting.
Yeah.
So the other question is, what do you generally tell a student or postdoc that want to work with you?
Yeah. So I think that science, doing the kind of research we do in the lab, is both, the most frustrating thing in the world and also the most rewarding. There are times when you are just going to want to pull your hair out and just leave the lab in tears because nothing is working and it doesn't make sense. And there's times when something comes together, or you make a discovery or you have a finding that fits with your hypothesis, and it's just, the most exciting, most amazing feeling. So it's sort of this roller coaster. And I think it's about trying to find a balance in there somewhere. Somebody once told me, so I think maybe this is the take home. Somebody once told me that your results are never as good or as bad as you think they are, which I think is true. And I think that's something that we sort of have to find that balance of in joining a lab and starting to do research.
Yeah, I think that those are really good advice, and it's very similar to what our scientists told me during this interview. So
yeah, there you go. Okay, good. Yeah.
So what do you tell them when they are graduating? I don't know if you already graduate students because we are kind of new.
Yeah, actually, I very proud PI of two recent master's graduates. So I've had lots of undergrads, but two master's students kind of come through the lab and graduate in the last little while. One actually just defended like last week.
Oh, perfect.
So not too many, but a couple of lab graduates. Yeah, I think there's sort of a fear once you finish grad school that you've spent all this time, maybe a 2-year master's or a 4 or 5-year PhD, that you've spent all this time like focusing on this tiny little research question. And I think people fear when they graduate that they don't know anything other than this tiny little thing that they've learned. And so I think the advice is that, the skills you learn in graduate school are so much bigger than that and apply to just all sorts of things. And I think, in graduate school, you learn to become like a really critical thinker, and you learn how to, answer questions and how to find the answers to difficult questions and how to juggle these complex problems in your mind, how to communicate, how to write, how to speak to different audiences, to scientists, but also how to explain to your mom and dad what you do, so I think you really develop like a lot of amazing skills. Even in a short master's degree, usually you end up mentoring undergrads. So you develop these leadership skills, independence, teamwork, it's sort of everything. And I actually don't think there's a lot of places where in such a short period of time you can get that much experience. So I think to graduating students, I think a good message is to remember kind of how much you have evolved and learned in this time, and that it's not just about your little tiny thesis project.
I agree. I think that that's the key message of the graduate school. You learn so many things. It's similar also to undergrad. We get so much information, but we cannot remember everything. At least
Yep.
I believe that it's a tool to know where to search for something, like.
Yes, exactly. Which I still do all the time and think about all the time.
Yeah.
Yeah.
So the last question is, what do you remember about your first CSM meeting that you attended?
So I came late to CSM. Maybe this is why I never thought of myself as a microbiologist, going back to the previous question. So I never attended CSM when I was in grad school. And then I went to the US for a postdoc. So I felt I was sort of disconnected from the Canadian research community. So the first one that I went to was actually right at the transition moment where, I don't know if it was at the University of Waterloo. I want to say it was 2018 or 17. We can probably find that out. We could probably fact check that.
My first one was 19, but I think that was 18.
Okay, so I think that was 18. So I was right. I think I had probably just accepted and was like coming back to Canada for this job now at the University of Guelph. So towards the end of my postdoc is when I started to make sort of a concerted effort to say, okay, I want to end up back in Canada. I want a research job in Canada. I want to get back into the Canadian research community. And that's when I actually started applying for all the Canadian conferences and CSM in particular. So I think that was my first one. And it was great. It's so great to be back in the Canadian research community, which I'm still very happy to be a part of, and have always found, well, subsequently, I guess I haven't been going for that long, but I have gone to every subsequent CSM meeting and have always found it to be just such a diverse and welcoming and dynamic environment, and have always found interesting new people to talk to and new collaborations to establish. So yeah, I've always really enjoyed that.
Yeah, I also agree. My first one was 19. Unfortunately, there was no in 2020. And hopefully,
right. Yep. That was supposed to be Guelph.
Yes, well, we think that we will keep it in Guelph. So that's good news.
Yeah.
Hopefully we can see each other in 2022.
Yep. Let's hope so.
Yes. But yeah, I think that the community is amazing. That's why I wanted to be involved, because it's hard to find societies that fight so much to showcase students, to showcase new professionals.
Yes.
Always keeping our amazing researchers involved.
That's true. And it more than a lot of conferences and societies really highlight students, I think gives students a lot of opportunities, and the platform, I can't remember what they call them, like talks selected from students, and
yeah, like the contest.
Yeah, exactly. And the awards. And I think students also always feel welcome there, like not like, I don't know, like at some of these huge, scary meetings that can be intimidating. So yeah, I think it's a great society and group to be part of.
Thank you very much.